Okay, guys, starting on chapter 9, talking about the way bacteria grow or microbes. Okay, the growth is, when we're talking about growth in this sense, we're not talking about an individual, we're talking about population growth. And this is due to reproduction of the individual microbes, same way it is with folks, right? These guys can either be in a discrete colony, which is one of those things that you see on the petri dish, or they can be in a biofilm. Now, the colony, we've said this in the lab, but in theory, each of those colonies comes from a single cell that divides and divides until it does that. A biofilm is kind of complex. If you've got lots of different types of bacteria, you've got a protective coating over it to maintain the environment. There is the ability for water to flow through. And biofilms actually are a hot study as far as the treatment of infectious diseases. Probably the biofilm that most of you are familiar with is plaque on teeth. And that's why we have to go and get our teeth cleaned every 6 months to get that plaque off. Okay, one of the main ways that bacteria are going to reproduce is binary fission. Here is the process. So the guy grows, right? Afraid our thing is out of alignment, but any case, there's the cytoplasm, the cell wall, and there's the chromosome. So we've got, this guy has got to grow. He's got to grow his cell wall, his plasma membrane, and he's got to duplicate his chromosome. When that happens, this guy starts to build a wall between these two guys. That's called the septum. When it gets completed, these guys can separate. And of course, since we're talking about population growth, and these two are going to make 4 more, which are going to make 8 and 16 and 32 and so forth. Now, this guy is showing that his time to go from 1 to 2 cells is 30 minutes. So every 30 minutes, this guy is going to double. So in an hour, instead of having 1 cell, we have 4. 2 hours, we have, I can't remember, 16, I believe. There's a photomicrograph showing bacteria, and no doubt these guys have got the septum, but they just hadn't finished it. DNA replication, septum, cell separation. And you guys can read that. There's nothing there that we haven't already mentioned. So obviously it's got, before it can divide, it's got to grow, and it's got to make sure that both of the daughter cells have the full complement they need to get along in life. Got to duplicate the DNA. On the circular DNA, there's something called the point of origin, or the origin, and that's where you start. It's actually, sorry guys, okay. So it actually goes both ways, duplicating the DNA until it gets finished. Opposite directions along the chromosome until it gets to the end. The center of these cells is going to constrict until the 2 cells are formed, and it's going to go through cytokinesis. Now over here they're talking about a protein called FTSZ. This guy makes a Z ring on the plasma membrane, not on the walls, plasma membranes, and helps to constrict it. Same thing as almost like a cleavage furrow in the human cells, and then as it brings it in, the bacterial cell walls start to grow, and that's our septum. Generation time, on the example we saw it as 30 minutes. There are some bacteria that double in 20 minutes. At the right conditions, they'll go fast. That's the generation time, or the doubling time. That's why we have a problem, because some of these guys can reproduce very fast, but our immune system takes a little while, up to 10 days, to mount a full response. So a lot of times these guys can overwhelm the defenders and kill you before your immune system actually is prepped and ready to go. The bacterial growth curve. So here's this the beginning. This is going to be called the lag phase. Why? Because it's flat, so we're not actually increasing. This is the numbers of living bacterial cells, not counting the dead ones. That's a logarithmic, and it gives us this chart. So once this bacteria has the right conditions to double, there's going to be a delay because this is time. There's that time delay there, and that's the lag phase. That is the time it takes to duplicate the DNA and to manufacture all the things that are needed. Once it starts, since we're doubling 2, 4, 8, 16, 32, 64, 128, we increase the numbers on a very steep rate. It doesn't take very much time for us to get a huge number of bacteria. This is called the log phase. That's an exponential increase in the number of living cells. Now, if these guys are in a particular culture or anything, or wherever they're at, if they don't keep getting fresh supplies of nutrients, and they don't have some way for the waste products to be carried off, then they're going to stop their increase, and that's going to be our stationary, the plateau. The rates of new cells dividing and the rates of cells dying are roughly the same, so you don't really have a net increase. And they're dying because basically there's no more nutrients and there's too much waste products, and basically the only increase of cell division during this process comes from the bacteria that are dying and releasing those nutrients when they die. The other bacteria get them. And then finally we have the death phase, and it's an exponential curve, only it's going down. And again, what we're seeing is even though there may be very few divisions, most of the bacteria are dying and not being replaced. During stationary, okay. And as the growth stops, the bacteria stop making more stuff to make more cells. Now, I got to tell you guys, this is where most of our antibiotics are going to have their biggest effect, is in the log phase. Why is that? Because these guys are building cell walls, and it's in the building of cell walls, not the destruction of already built cell walls. That's where most of the antibiotics we use work at. We sabotage their new cell walls. We don't actually damage the ones that they already have. So stationary cultures, these guys, they're not going to be quite as susceptible because they're not building as much. If these guys can make those endospores, those armored tanks, at this phase, many of these guys, since they're being stressed by the lack of nutrients, etc., many of these guys will start forming spores, sporulation during this time. Now, if this guy is helped along with antibiotics, then this is where we get some drug resistance. If we've got 100 bacteria, for example, and I kill 98 of them with the penicillin, and those other 2 are resistant to penicillin, then I'm going to kill these guys, and then these guys are going to start 4, 6, or 8. Do you see what I'm saying? This is those persisters. These guys don't respond to the same antibiotics, and they're just going to start this whole curve over again. So this is a problem, and that's one of the reasons this growth curve is important. This is where we want to hit them at with the antibiotics. And a lot of times we'll use 2 antibiotics so that if, you know, maybe antibiotic A kills 98 and leaves 2, antibiotic B kills 97 and leaves 3, but guess what? They're not the same. These 2 are killed by B. These 3 are killed by A, so it doesn't matter. The net result is all 100 are gone. Sustaining microbial growth, a lot of times they want to keep a culture going on, especially since we use bacteria to make things like insulin for us. So in order to get away from that death curve, what we do is we send in new medium with new nutrients, although limited. We let air come in here, and because we have air in here, we're going to have some pressure, which is going to take an overflow, and that's going to take the waste out. So even though the same bacteria won't be there, we'll basically keep the whole culture growing. Chemostat maintains it in whichever phase, growth phase. The open system, you have to put this fresh medium and you remove the old stuff, which just makes sense. You've got to give them food and you got to remove the waste products because otherwise they'll become toxic. These guys can be studied then at these nutrient levels, and they use them for several things. I'm not really familiar with that because that's not healthcare related, but anyway. How do you measure how many bacteria you've got? Those guys are tiny. Here's a method that you don't have to incubate. Incubate is plating them and putting them on a plate or in a tube. Microscopic counts. There are ways to count these directly through a microscope. They've got slides that have a grid pattern on it. I'm thinking this is probably old school, and I seriously doubt that there's very many, if anybody, who does that nowadays. Stained prokaryotes and large eukaryotes. This is probably the way that most of the labs are going to work. They're going to have an electronic counter. They count the cells because they interrupt an electric flow that are flowing in front of the electronic detector. So every time a cell passes by there, it's like it blocks the light. That would be kind of this is an analogy. It's not perfect, but if you wanted to count people and you had a beam of light shining, kind of like you do with those bells that they have like on stores and things. Every time someone walks through there, it's going to interrupt it. And instead of going bing, it's just going to add one to its counter. Okay. So this is kind of sort of the way these guys work, that culture. Flow cytometry, you do actually use light, and you've got a receiver and you've got a light source, and every time these guys pass, they interrupt the light. That's recorded, and it advances the counter. Now that's the ways that is way without actually growing a culture, incubating them. Now, we've got these methods that do require serial dilution, viable plate counts, we'll talk about that, membrane filtration, and most probable number. This is serial dilution. This is actually one of the labs that we do when we can. So I take 1 milliliter of the original culture, from a tube or from a plate, doesn't matter. I add 9 milliliters of broth. 9 and 1 is 10, okay? So if the original culture came from a 10 milliliter tube, I've got 1/10 of that, so that is diluted 1 to the 10th. I let this grow, and then I take 1 milliliter out of that, so that is 1/10 of 1/10, and that is 1/100th. You guys see what we're doing? By taking 1/10 of this one, we're actually diluting these. Now what we're going to do is we're going to take 1/10 of a milliliter and we're going to put it on the plate, do the streak plate. And then we're going to incubate these guys, and then we're going to count. Usually these guys still have way too many bacteria, so they're going to be too many colonies. Somewhere around here, it depends. You're going to get a number you can count. You can estimate the bacteria from the number of colonies, and of course here, this is 1/1000. There actually apparently were no bacteria in that loop. It's just too diluted. This is one we used to do too. We put the bacteria in a tube, test tube, let them grow. A lot of times the bacteria will make the fluid cloudy when they grow. So what we do is we pass a light through there again to a detector. I guess I'm coming the wrong way. I need to go this way. And the detector is going to say, what we do is we first measure it with perfectly clear fluid. In other words, that's the growth medium without any bacteria at all. And then we put this guy in here and we check and see how much light still gets through. The amount of blockage is going to give us an estimate of how many bacteria are there. The only problem with this guy is it's going to count dead as well as living bacteria, because even a dead bacteria, as long as it hasn't, come apart, it can still block light. So that's called turbidity. As a practical matter, when you look at the patient's Foley bags, that is also one of the things we're looking for, is turbidity, to let us know whether our patient may have a urinary tract infection. The indirect, the turbidity, metabolic activity by measuring how much nutrients are used, how much waste is produced, or the pH, and we'll do some of that later on. Dry weight, you filter these guys from their liquid media, and you weigh them. It sounds like it's fun. And genetic methods, not all bacteria can be cultured, by the way. You just have to get DNA samples to kind of give an estimate. Biofilms, we talked about that. Reversible attachment. Planktonic cells are just cells that attach themselves to a surface, such as your teeth. These guys eventually they glue themselves there. They're irreversibly, and this can be done in minutes. They grow, they divide over the next little bit. These guys are going to form this EPS, and they're going to form these water channels. And then eventually these guys are going to have groups that leave and go and colonize new sites. Surfaces, medical devices, mucous membrane, they have something called quorum senses, where these bacteria release a molecule, and once there are enough of those molecules, that's when they start to form this protective biofilm. It's sort of like a slime coat. It protects them. Dental plaque, we've already talked about that. Right now, this is a big hot topic in disease treatment. A lot of people who get infections through cuts and everything have biofilms. The problem with these biofilms, guys, is it's hard for antibiotics to get in there to the bacteria. And if you're not there if you can't be there where the bacteria is, then the antibiotic is no good. Other factors, oxygen, temperature, pH, and water. We've kind of talked about temperature and pH, but let's talk about the oxygen. Oxygen is variable. Oxygen is extraordinarily toxic. But we, for instance, have a lot of enzymes that detoxifies oxygen. Some bacteria have a lot of enzymes to detoxify oxygen. So they are perfectly happy and at home in an oxygen atmosphere. Some of them have none, And so they will be destroyed by oxygen. Those are anaerobic bacteria. And then you've got several groups that are in betweens, and we'll talk about those in the next few slides. Obligate aerobes, these guys cannot live with oxygen. They would die without oxygen the same as we. Obligate anaerobes, which I talked about, if they're with oxygen, they will die. And we talked about the oxygen, some of the superoxide radicals. These guys are very destructive to any other molecules that are around. You start messing up with molecules, you start causing DNA damage. That's when they were thinking, and that radical oxygen, or these free radicals, you may have heard that term, might be a causative factor in the development of some cancers, maybe. These guys are oxidizing agents. They're going to steal electrons. Some places you don't want to steal electrons from because these are important molecules and they're essential for the life of the cell or whatever. Irreparable damage. Singlet oxygen. Oxygen, if you guys remember, has to be bound. They share each share a pair of electrons. This is one, and this guy needs 2 electrons. He's got 6 electrons and of course 6 protons, so he's balanced. But he is electron hungry, so he's going to grab stuff. Superoxide radicals, these guys, when these guys aren't completely reduced, they get this. We also have the peroxide anion. These guys, we use superoxide dismutase. That's one of those enzymes that we have to make this harmless. The peroxide anion is simply, you guys know, peroxide is H2O2. So this guy has got an extra oxygen, and there's really not enough electrons to go through there. So we have something in our cells called catalase, and it will take this guy and turn him into oxygen and water, which are both relatively harmless. Some bacteria have that too. As a matter of fact, one of the things we do in the unknown is take and put hydrogen peroxide on a slide with the bacteria. If you see the bubbles, you know it's producing oxygen, and that means it's catalase positive. If you don't see it, it's catalase negative. Hydroxyl radical, this is that OH. Again, this guy is really hungry for a hydrogen, so got to find one. And again, that's due to hydrogen peroxide not being completely reduced. So we got these obligate aerobes, and we've got the obligate anaerobes, and they know what they like. Now we have facultative anaerobes. What does that mean? It means they prefer oxygen, but if worse comes to worse, they can survive without it. Okay? These guys are aerotolerant anaerobes, so they prefer not to have oxygen, but if it's present, they can tolerate it to a certain degree. So in a sense, these two guys are opposite. These guys prefer to be aerobic, but they can do without. These guys prefer to be anaerobic, but they can be okay with it. And then you have these microaerophiles. These guys are okay with oxygen to a certain degree, and then it's because they have enzymes, but they don't have a lot of enzymes. So they can deal with low concentrations of oxygen, but they are just as susceptible to being killed as the obligate anaerobes if they have too much. They'll just be more than their small stock of enzymes can handle. Okay, temperature. We've already talked about this when we were talking about that earlier. That there's a growth curve. So there is an ideal temperature for all these bacteria. You guys remember in the lab where I think it was the ubiquitous microbes. If you get a microbe and you get it off the surface, then this ideal temperature is going to be room temperature, somewhere around 72 to 75, whatever. If you get it off a person's hands, then it needs to be about 37 C or about 98, 99 Fahrenheit. So all of these bacteria, all the bacteria don't have one range. They all vary wildly. And it's because of the 3D proteins. We've talked about that. Lipid-containing membranes of the cells and the organelles are also temperature sensitive. The temperature increases, you can turn salad fat into oil liquid, right? If it's too low, these guys get kind of get hard, and they become fragile. If it's too high, they melt. Here's the temperature range. You've got these psychrophiles. Does that mean that these guys are like Silence of the Lamb? Not really. But look where they're at. They're at about 10 degrees Celsius. That is way below body temperature. I wonder what that temperature is. Well, that's just about around the temperature of your refrigerator. Because of these guys, you can't put something in your refrigerator and have it stay good forever and ever. A week at the very most, and your stuff is going to start looking funny and smelling funny, and you don't want to taste it. The food in your refrigerator will need a shave because it'll have green beard growing all over it, or black, or whatever. But these are the guys that are adapted to lower temperatures, so this is their preferred range. They like it there. Then you've got the mesophiles. These are the guys that are going to be the ones that we typically think about. These are the ones where the human pathogens, because these are the guys that are going to be room temperature to body temperature, maybe a little bit more. So these are kind of the middle guys. These are the guys who like it hot. And by hot, I mean just they go all the way to 80 C. Can't remember how much that is in F, but it's over, that's 212 F. So that's probably going to be like 150, 160 degrees Fahrenheit. That's warm. These guys, and these are basically going to be archaea for the most part, hyperthermophiles. They like it to be boiling hot. Now these are the growth curves, guys, so you can tell the high point is the optimum temperature. And again, these are for classes of bacteria, so each individual bacteria may move around that, but basically that's a good generalization for the class. pH, same thing. We talked about those. Neurophiles, these guys are good around pH of 7, which is neutral. And our pH is 7.35 to 7.45, so it's about right there. So these are going to be the guys that are going to be human pathogens. There are some, though, that can be pathogens in this range in our stomach. These guys are acetophiles. They like low pH. These guys are the alkaliphiles. They like the high pH. So there are niches for every living thing, and these guys fill these. There are a lot of we talked about the waste products, but most of the bacterial waste products we haven't spoken of yet is going to be in the form of acidic waste. And these guys can actually accumulate and damage these neutrophils. But the acidophils will be very happy to live in that acidic environment. The alkalinophils, they live in environments that are naturally alkaline, soil and water. Water. All biochemistry is wet chemistry because all biochemistry requires water in order for the reactions to be done. So just like us, bacteria need water. If there is no water, then they will die. That's why desiccation, drying out things, is an effective way of killing bacteria. Um, Some can survive longer than others because they've got special water conservative structures, but and in the endospores and cysts and the eukaryotes, they're more like, again, like a lifeboat, and you basically put just the bare minimum of cell components in the DNA. Water, though, can have two effects. There's osmotic pressure and hydrostatic pressure. The hydrostatic pressure, water weighs about 8.5 pounds per gallon. So if you're at the bottom of the ocean, guess how many gallons you got on top of you? If you were standing right there, that was your head, I would say there's telling how many thousands of gallons, but the weight of that would be like 10,000 pounds on every square inch. I don't think our skulls would hold up under that. But again, there are bacteria, especially archaea, that love it down there. As a matter of fact, they can't live without that hydrostatic pressure. Osmotic pressure, if you remember, we were talking about. The reason that our antibiotics punch holes in the bacterial cell walls is because by doing that, since there's a lot more concentration of certain proteins in there, you will have water flow in and cause the bacteria to lyse, to go pop. But you can't make the bacteria lyse really once inside that rigid wall. The water will flow in, but then the pushback, because eventually it's got so much pressure in there that it's not going to. But if that wall were gone, then water would continue to flow in until it pops it. Osmotic, hypotonic, hypertonic. So we're saying that that in many ways this is going to be hypotonic compared to the inside of the bacteria. If you were in a hypertonic solution, then the wall really not going to be that valuable because you're going to crenate, you're going to shrink because the water will flow outward. And again, these are going to determine where these guys live. And we have obligate and facultative halophiles. An obligate halophile cannot live unless it's in a very salty environment. Ocean, Great Salt Lake, Dead Sea, think of that. Then we have facultative halophiles. Our skin is salty, and a lot of bacteria, they get on there, and guess what? We suck the water out of them and they die. But there are some that are perfectly fine with that, and amongst those are the Staphylococcus bacteria. Staph aureus, the pathogen, Staph epidermidis lives on our skin. It's part of our normal flora. We already talked about that. Those barophiles are the ones that live at the bottom of the ocean, and they're fine. As a matter of fact, without that, their enzymes would lose their shape and would be no longer functional. Their enzymes depend on that. Oh, the Great Salt Lake. I saw that when I was in Utah. That's nice. Real blue. As far as the nutrients these guys need, usually when we inoculate these guys, when we do our streak plate and put them on the agar, which is our media, those agar contain enough nutrients to get the bacteria to grow for a time. Of course, eventually they're going to run out of nutrients, but that's fine for our purposes. we get these guys from different places, environment, clinical specimens. When we do a wound culture, we get bacteria from there. Some they're just coming from stored specimen. In the labs, we actually have plates that we keep. We just put them in the refrigerator and slow down the process, and it helps to prolong the life of the bacteria. Culture, that's when we put them on the plate, or the broth media. Most prokaryotes cannot and have not been grown. Nutrient broth is pretty common. We put them in a test tube in that yellow broth. Agar is complex polysaccharide that comes from red algae, and it's actually a liquid, and it gels up and becomes a solid surface for the bacteria. And the bacteria can't eat that agar, so it stays, the bacteria stay on that plate. And it really helps us with our colony morphology. 6 types: defined media, complex, selective, differential, anaerobic, and transport. We'll defer that to the lab because we have a lab that talks all about that, and I think that'll be a better time. Some of these selective media, we have some things that only gram-negatives can grow on it. Gram-positives won't like it. We've got differential media like blood agar. That's actually got sheep blood. They've streaked 3 different bacteria on there. That one did nothing. That one totally did away with those red cells, completely metabolized them. This guy is somewhere in between. See that greenish haze? He partially digested those red cells. These guys measure acid production. Remember we talked about acid being a main metabolic waste? So we have a pH indicator, and we've actually got some that are yellow and they turn red, some that actually kind of become yellowish. And there's an anaerobic chamber. This is a place they've got things in here that actually take out all the oxygen, and so this becomes depleted of oxygen, which for anaerobic bacteria is what you need. Okay, let's see what we got. McConkey agar is selective and differential. See the Staph aureus on the nutrient agar? That's like TSA plate. And on the McConkey agar, it won't grow at all. But E. coli does. Why? Because this is selected for gram-negatives. Okay. Now, in the gram-negatives, the Salmonella enterica and the E. coli both grow on this gram-negative, but look at what happened to the Salmonella. They actually digested some of the nutrients on there that the E. coli could not use. So anyway, but guys, we'll talk more about that in the lab. And that winds up this chapter. Appreciate you hanging in there with me, guys.